and welcome to OsteoBites. My name is Christina Iptoma, and I am mom to Osteo Angel Dylan and the Director of Scientific Programs for MIB Agents. And today on OsteoBites, we are talking with Dr. Bilal Omer about armored CAR T cells for osteosarcoma, a phase 1 trial of GD2 CAR T cells combined with a novel interleukin-7 signal. Thank you so much, Dr. Omer, for joining us on OsteoBites today. We're thrilled to have you, and thank you to Matt Allen for joining us as a panelist today. Matt is an MIB Agents Junior Advisory Board member and Osteo Angel sibling. So Dr. Bilal Omer is a physician at Texas Children's Cancer Center. In 2013, he completed his fellowship training in pediatric hematology-oncology. He's a member of the Bone Marrow Transplant stem cell transplant team and medical director at the Center for Cell and Gene Therapy at Texas Children's Hospital, Baylor College of Medicine. He's the principal investigator on 4 clinical trials for cell therapy. And so last week we had Dr. Breland Wilkie, who gave us a great overview of the immunotherapy landscape for osteosarcoma. And Dr. Omer's work popped up a couple of times during that conversation. So it's great that we can do a deeper dive with you today. Before we get started, I do have a few reminders. We have a little over 4 weeks left until our Factor conference. So register now. We'll put all the information in the chat with all the links to get more information. We do have a couple of exciting updates. First, the detailed agenda is now up and available, so you can check out the different panels, the speakers, and the presentation topics. You'll see some really cool programming in there, like Sound Bath and Healing Touch, which are things you probably won't see at a regular scientific conference, but these are part of a new wellness track that we're adding this year that may be of particular interest to families that are in treatment or bereaved families who may need a break from the scientific sessions. So check those out. It's another reason to come. And then secondly, we just opened up virtual registration. So we'll be live streaming the scientific sessions of the event. So if you aren't able to travel, you can catch the scientific sessions either streaming live or after the fact on demand. Nothing beats the energy of being at Factor in person and connecting and interacting with the colleagues and seeing all the patients and families in person. But virtual is a great alternative if you can't travel to San Diego this year. And lastly, before we get started, I want to thank our sponsor of this episode, BTG Specialty Pharmaceuticals. BTG provides rescue medicines typically used in emergency rooms and intensive care units to treat patients for whom there are limited treatment options. They are dedicated to delivering quality medicines that make a real difference to patients and their families through the development, manufacture, and commercialization of pharmaceutical products. Their current portfolio of antidotes counteracts certain snake venoms and the toxicity associated with some heart and cancer medications. Their drug Veraxase is for high-dose methotrexate toxicity. All right, now Matthew, would you like to introduce yourself?
Sure, thank you, Christina. My name is Matthew Allen. I'm a member of the Junior Advisory Board for 2021 and now 2022. I'm the brother of an Osteo Angel, and I've previously been involved with MIB doing gamer agents, and yeah, I'm really excited to be here today. So I'll throw it over to Dr. Omer to get started.
Thank you so much for the kind introduction, and thank you, Matthew, for being a panelist. So feel free to interrupt me with like any questions. So I don't mind at all. So this is can be a little bit complicated. So feel free to kind of stop me at any time if it's kind of not clear or something. If you want something explained. So kind of starting with some basics. So we use T cells kind of as a platform for our treatment. So if you kind of look at blood, there's white blood cells, and there's platelets. So in the white blood cells, we have immune cells, and part of these immune cells are lymphocytes. And lymphocytes are responsible to fight infections, especially with viruses. So certainly in the context of COVID, a lot of you probably have heard about kind of T cell response and B cell responses. So B cells make antibodies, but then T cells are able to directly target cells that are either infected with the virus, for example, or for example, abnormal cells that are becoming cancerous. So T cells can actually directly kill these type of cells. So T cells are a very important part of our immune system. And in a way, they're kind of like a, like a smart part of the immune system. So there's some other cells that are just kind of, they look for very kind of basic kind of foreign antigens, but T cells are very sophisticated. They can interrogate cells. They can recognize, millions and millions of antigens, and they can, and then they can kill target cells in a controlled manner. So without causing like usually kind of excessive kind of tissue damage. So there will still be inflammation, but there wouldn't really be any kind of tissue damage. So we kind of take advantage of this. So we actually arm these T cells with a chimeric antigen receptor. So we can build these receptors. So there's a recognition area that can actually bind to a target. So we would pick a target that's expressed on a tumor cell. And then once the recognition area binds to the tumor cells or the target, and then kills the cell. So this is a very artificial type of receptor. So it's not something that is actually really present in nature. So it's a pretty artificial system. And what it essentially does is kind of turn these T cells into these killer cells. So when they see this antigen, they just kill the cell. So they don't really care about this is like a friendly cell or tumor cell or infected cell, not infected cell. So when they have this CAR receptor, they just kill up that cell, and no questions asked, essentially. So you have to be very it's very important that you kind of pick a good antigen, because otherwise they're going to start killing off healthy cells, because they don't once they have this CAR receptor, they'll kill off any cell that has the target. So in the leukemia settings of blood cancers, we've been doing this for years and with very good success rates. So there's a number of FDA-approved treatments now using CAR T cells, and the response rates are pretty high. So, like for example, for acute lymphoblastic leukemia in children, so most patients who get these CAR T cells are multiply relapsed, and they have failed a lot of other treatments. So when we give these CAR T cells, we get response rates of about 80% of these patients achieve a complete remission. And then about half of these patients, so maybe about 40% actually don't need any other treatment. So if you just give a single infusion of CAR T cells, these patients do get a lot of inflammation, but then after the inflammation dies down, about 40% of these patients are completely cured of leukemia. So in theory, this is a very compelling treatment, because essentially you can do like one infusion, and potentially this can kill all of the tumor cells. And then these CAR T cells can actually they can persist in the body for a pretty long time, and usually they kind of go up, persist for some time, and they kind of start going down, and they can actually patrol the body for months, and look for any kind of remaining tumor cells, we kind of take advantage of that by again in the leukemia setting, and by sometimes just doing like a single infusion. So this is kind of like how we make these T cells. So first we take a sample, blood sample from a patient, and then we separate the other blood cells from the T cells. Then we introduce the DNA with the CAR gene with the patient cells, and we actually use like inactivated viruses to accomplish that. And then once we put the DNA in the T cells, we grow them in the lab until we have enough cells. Depending on the indication, we usually make a couple 100 million cells, and then we kind of freeze them down and kind of use them for some time. And they actually stay stable in the freezer for years.
Dr. Omer, I actually have a question for you about that. I know some other CAR T trials will require apheresis to acquire the sample. And so can you talk about the difference between, with this trial, it's actually pretty easy to just send a few vials of blood in the mail versus having to do apheresis. So can you kind of explain maybe kind of the difference between those two different collection methods? And
yeah, sure. So apheresis is like a way to kind of get like a lot of lymphocytes at the same time. So you can do apheresis, and you get like a huge amount of T cells. You kind of use them in the lab. So if you just do a blood, if you just do a blood draw, you don't get as many T cells. We definitely get less T cells from an apheresis, but we actually get very good expansion of our CAR T cells. So even without apheresis, we can actually really get high cell numbers. We actually don't infuse super high cell numbers. One reason is I'll talk about the cytokine gene in a second. So I think these, even at lower cell numbers, we actually get very good expansion and kind of very good, potency. So we don't need like the really high kind of starting T cell numbers. So for that reason, like you said, we actually don't need the apheresis. So what we actually do for most patients, we collect 60 ml of blood, so about 2 ounces of blood. The blood is often shipped to us. So we receive the blood, we check the T cells, can make sure they look okay, and then we can freeze them down, and then we can actually make the CAR T cells at any time after that. There's some concern in the field that so if you grow the CAR T cells for too long, then they start getting tired and exhausted. Initially, people used to grow these CAR T cells for 2 weeks, and we can get like a huge amount of T cells. But I think there's increasing concern that if you grow them too long, by the time you infuse them, they're already kind of at the end of their lifespan. So the goal right now is to kind of shorten the time of manufacture. So our manufacturing time is about 1 week, which is kind of in line with what people are at the moment trying to accomplish. It is possible that maybe in the future we'll say, well, maybe 1 week is too long. Let's kind of go for 5 weeks, and in that case, potentially might have to go back to apheresis. But currently, our 1 week manufacturing time is actually really good and kind of in line with like what most other trials do, like the kind of more newer, kind of more modern trials.
Great, thanks.
Sure. So going to like the signals. So there's like 3 signals that the T cells need. So signal 1, signal 2, and signal 3. And I'm not going to go into too much detail about this, but so the very initial CAR T cells just had signal 1. So they just were able to kill the tumor cells, and while they killed the tumor cells, there wasn't really any kind of long-term success with this. So all of the standard CAR T cells now that are FDA approved, and there's a number of them now, they all have signal 1, and they all have like a signal 2 as part of their gene. So if they recognize a tumor cell, you get the signal 1, and then you get signal 2, which is called a co-stimulatory signal. These 2 signals seem to be enough to actually, get the good results that we've seen in the leukemia and blood cancer setting. What they don't have is signal 3, and the thought is you don't really need signal 3 because we give chemotherapy before. The chemotherapy causes a lot of inflammation. Inflammation means a lot of cytokines. So there's a lot of cytokines kind of floating around in the bloodstream. Leukemia cells are in the bloodstream. So as soon as the CAR T cells are infused in leukemia, they see all the cytokine from the chemotherapy. So people don't really worry about cytokine signal 3 in the leukemia setting, because like the thought is like the cytokine is floating around because like we make it we increase the inflammation from the chemotherapy before, so we don't really have to kind of provide that extra cytokine. But because of that, all of the CAR T cells that are currently FDA approved, they all use what's called lymphodepletion. So like they all use the same type of chemotherapy, which is cyclophosphamide and fludarabine before. But what we think is that solid tumor is a little bit different. So we have osteosarcoma is a solid tumor. So that means like there's like a big chunk of tumor cells that's sitting somewhere in a bone or somewhere else in the lungs. So the problem is like when the tumor cells get to the actual tumor, there's not a lot of cytokine around. So there's like a lot of data that these tumors have a way to kind of change the environment. So when the T cells get there, there's not much oxygen. There's not a lot of nutrients, and importantly, there's like very little cytokine. So like I said, in leukemia, there's like plenty of cytokine floating around. And the tumor cells are just floating around. So we don't really have to worry about that too much. But in solid tumors, it's different. So the T cells get there, but they don't have the signal 3. So they're not completely activated. So what happens after a couple of days, they essentially die off. So we were thinking when we started this project that there's just not enough signal 3 for these CAR T cells, and we have to figure out a way to provide this this signal 3. As I mentioned, so this whole CAR receptor is engineered. So we thought we can engineer cytokine receptor in a manner similar to like this CAR receptor. And when we initially kind of started working on this, there wasn't really like a term for this, but recently people have started using the term armored CAR T cells for this. So like CAR T cells that have this extra signal 3 are called, some people now call them armored CAR T cells. So there's a number of different cytokines, and T cells don't necessarily have to have all of the cytokines. So there's some key cytokines that are most beneficial to T cells. And we think one of the most important one is IL-7, so interleukin 7. And the reason that we think it's very important is like it can help both younger and older T cells. Some of the cytokines are really just good for young T cells. Some of the cytokines are really just good for old T cells, but IL-7 is actually good for all of the T cell populations. And then secondly, so if you expand T cells quickly, so if they grow very quickly, if you have other cytokines, they don't prevent T cell death. But if you have IL-7, even after they expand a lot, these T cells can survive longer. So that was a second reason for us to look at IL-7 as a potential cytokine to provide the signal 3. So we focused on like an IL-7 mutant. So this gets a little bit technical here, but we essentially using like a slightly mutated IL-7 receptor. And the reason for that is usually IL-7, once it provides signal to the T cells, it goes inside the cell really quickly and stops working. So if you want something that works for longer, you have to change it a little bit. And we mutate it so it can actually stay on the T cell surface and not disappear after a couple of hours, a couple of days. So we use the IL-7 mutant that essentially always kind of keeps like the interleukin 7 activated. So that means you always get signal 3 to the T cells. You always have interleukin 7. So you always have like enough essentially fuel for the T cells as long as the tumor antigen is there. So like I mentioned, so the T cells need signal 1, signal 2, and signal 3. So the thought is as long as there's like signal 1, which is like the tumor, these T cells will have enough drive to kind of keep expanding and keep surviving, because like now they also have the signal 3. And then we made a couple other tweaks to this interleukin 7, so we can easily detect it in the bloodstream, so we can so that we can kind of track these T cells and kind of see like how much of the interleukin 7 is on the cells. So generally, we try to minimize, animal experiments, where I think all of us are very kind of conscious of, trying to kind of use as little animal experiments as possible. But, before we actually put something in a clinical trial to test in patients, we have to do at least some animal experiments. And so we tested this C7R, we call this C7R signal 3 gene. So you can kind of see here, these are mice here. These are like this is the survival curve. And you can see if you just have a regular CAR T cell, you can see that the tumor is controlled a little bit, and then these mice, do survive a little bit longer. But then with the C7R, you can see that except for one mouse, all of these mice were actually cured. So that was pretty encouraging to us, and we had a couple different mouse models where we tested this, and we kept getting the same results. So with this encouraging data, we did we did apply for a phase 1 clinical trial. So essentially, this is like the outline. First, of course, you have to make the product, so that takes a little bit of time. The actual manufacture of the T cells doesn't take very much, as I just mentioned, so just a week. But coordinating the blood draw takes some time, and then we do a lot of testing. So the testing can take anywhere from like 2 weeks to 4 weeks, depending like, if you have to repeat anything. And then for some of the patients, we did the lymphodepletion. So this is the standard chemotherapy that people tend to get before all the CAR T cells. And then we infused the T cells, and we actually tested a couple different doses. An initial dose, we actually tested the CAR T cells without any kind of chemotherapy. And then we had 3 dose levels where we're testing these CAR T cells, 10 million cells, 30 million cells, and 100 million cells. And then, of course, then after that, we can follow the T cells and kind of see how they expand. And then, of course, at one point, we want to see, is there any type of response in terms of the tumor. Some results from the clinical trials in terms of the manufacture. So like I said, we are able to manufacture these T cells in about a week, but we do have to do extensive testing. So far, we haven't had any failures in terms of making the lines. And like we talked about before, we don't need a lot of blood. We just need about 1 ounce of blood for the manufacture, and then like 1 ounce of blood for testing of the blood for various pathogens. And then we use double transduction. So that means we actually have 2 different genes. So we have 2 different steps where in the first step we put in the CAR gene, and then in the second step we put in the cytokine gene.
Dr. Omer, just another question for you about the manufacturer, because I think this is somewhat, I don't know how unique this is, but at least of the trials I'm familiar with, unique to your study. So not only just the collection is a little simpler from the patient perspective, but also I think the trial had been broken out into the collection phase, collection and manufacturing phase, and then the actual therapy phase. And so I know just from personal experience, we were able to send in sample for manufacturer much early on, like before we actually needed to start therapy, which was fantastic though, because then when we needed to start therapy, the product was already ready to go. So that seems somewhat unusual, again, from what I've seen. So I'm just curious how you're able to do that. I mean, it also sounds like it's kind of a budget issue, because I'm sure it's an expensive endeavor to manufacture, and with not knowing for certain that they will get used. Yet it seems like there is a lot to learn from that whole testing, the manufacturing piece of this trial. So I don't know if you could just address that, because I actually think it's a great way to really increase enrollment, honestly, because you're able to get that sample collection easy. It could be done more easily up front, so that by the time a patient is ready to actually start therapy, everything is ready to go. Because as you know, oftentimes when a patient's disease is advancing, like they may not have, 2-3 months to kind of get all the stuff going. They kind of need to start therapy right away.
Yeah, I think you're hinting at some really, really important kind of points. And I agree. I think having that flexibility, I think, is helpful. Like I said, I think there's like a budget factor, because making these lines probably costs $15,000-$20,000 kind of for each line. So if you make them, it's a lot of work, a lot of cost. So on the other side, I think it's it's much easier probably to treat a patient who has like a lower disease burden. In osteosarcoma, we don't have as much experience yet. With neuroblastoma, we have a lot of experience. And in neuroblastoma, it's very clear, patients who don't have that much disease, they respond much better to CAR T cells. So if you like smaller tumors, like talking about like 2-3 centimeters, like an inch, and those patients can have complete responses, and or very good partial responses. Once you get to a point where you have like very disseminated disease, or very bulky tumors, very big tumors, I think it's just much harder to kind of respond to CAR T cells in general. So yes, I do think it's potentially like having that option to kind of say, well, right now the tumor is very small, or maybe like even in remission, or not barely detectable, and we're kind of trying something else. Making the CAR T cells in the meantime. I think that's that's actually, I think, a pretty promising approach, because like then you can treat a patient who has like smaller tumors, and again, like, with a probably like higher chance of success than like if someone was like, big bulky tumor. If you think about, if you think we are infusing like 10 million CAR T cells, but if you think about like a big bulky tumor, there's probably billions and billions and billions of T cells. So like each, CAR T cell has to kill off, thousands of tumor cells. So if you have a much smaller tumor, the amount of tumor cells can be a lot lower, like even less than that. So yes, I do think it's probably a good thing to, if there's a high chance that these cells are going to be needed at some point, to kind of think about this relatively early on, instead of kind of waiting till like kind of the last second, when the tumor is rapidly growing and it's very big. So yeah, I think that's a very, very good point. So another thing we kind of started doing is like, we initially didn't require any testing for osteosarcoma. We know that essentially all of the neuroblastoma tumors we tested are positive for GD2, and that we assumed that the same was true for osteosarcoma. And it is not that easy to test for GD2. Like it took us a while to kind of get like a protocol going where we felt we got pretty reliable staining results. So early on, we were not testing all of the patients, and then once we had this protocol going and we're pretty convinced it's working well, I started testing all of the osteosarcoma patients, because I think it's important to kind of at least like have like some idea kind of what's how much E2 is actually on the osteosarcoma cells. And so we actually found that there's actually a significant proportion of osteosarcoma patients actually are negative for GD2. So about 1 in 3 of osteosarcoma patients are negative for GD2. And then the remainder of patients, there's a little bit of like a range. And most patients have GD2 on almost all of their tumor cells, but that's not always the case. There's a smaller proportion of patients who only have GD2 on like a subset of their cells, like 50% or 60% or 70%. There's a possibility that some patients actually, some of the G2 can be washed off. So there's potential to kind of get maybe slightly lower results than are true. So the potential kind of for sometimes getting kind of false negative results. But we've been getting pretty consistent results with the staining. So while I think it's possible, I don't think that's like the main reason why proportional patients are negative. So what we've been doing recently is in the first step, we actually do screening for GD2. If it's completely negative, then we wouldn't treat. If it's kind of somewhere in the middle, then I think it's like a matter of kind of discussing. So are there other options available? Are families still willing to try it, even though there's a proportion of the tumor cells that seem to be negative for the target. If the expression is really high, then at least like that part we're kind of pretty confident. Okay, like we at least know that the T cells can target and kill off the tumor cells, and the expression is generally a little bit lower in osteosarcoma than neuroblastoma. So there's like two components. Like one is like what proportion of the cells actually have the target, and the second is like each of the tumor cells, how much GD2 do they have? So in general, neuroblastoma has more GD2. So it's probably a little bit easier for CAR T cells to target it. But I've done quite a few experiments, and it seems that CAR T cells can kill osteosarcoma cells just fine. But I think there's a potential question that maybe they don't get as much of a boost on like the killing, because the G2 is maybe not as high in all of osteosarcoma. So a couple of questions that are kind of still outstanding. I think in the future, I think there's a couple of agents that I'm working on that seem to be increasing G2 expression. So this is kind of relatively early on. So I don't want to kind of give you data that's not kind of confirmed yet. But there's a couple of classes of drugs that are already clinically approved that seem to be boosting G2 expression. So something that might potentially be something we might be using in the future.
And is there a correlation then between GD2 expression and response? Are you able to?
It's hard to know. Like we've had a couple patients, and so we test, we don't require post CAR T cell biopsies, so we don't have like biopsies on most of the patients. We did get them on a couple patients, and so it does seem like there seems to be like a higher proportion of negative cells in like the post infusion sample, and like the kind of really high expressing G2 tumor cells are gone. So it's possible. But I think in general, I think you probably want at least like 50% of your tumor cells to express G2. I think if it's less than 50%, then again, we don't have like a formal cutoff in the protocol, but I personally would probably strongly kind of discourage people to kind of do this if less than half of the cells express G2. I think if it's more than half, I think then I think we've been getting clinical responses. So again, like we don't have enough data yet to know for sure, but I think I'm pretty convinced that there's there's a proportion of osteosarcoma cells that have very little G2, but most osteosomas to express enough G2 to actually get killed by the CAR T cells. Okay, so that's kind of the testing part. So kind of getting to like the treated patients. So we had like a mix of neuroblastoma patients. You have two different arms. One was patients without lung mets, and those are essentially like most of the neuroblastoma patients. And then I think all of the patients, all of the 8 osteosarcoma patients actually had lung metastases. So all of the lung metastases patients were actually osteosarcoma patients. And then we had one patient who actually was a rhabdomyosarcoma patient who actually was G2 positive. And we had a mix of male and female patients. And like I said, all of the osteosarcoma patients and the rhabdomyosarcoma patient actually had lung metastases. And then median age was 14, and most patients were kids, but a couple of patients were young adults. So I think the most interesting data point to me, I think, is like the CAR T cell expansion. So here on the left, so this is data from like our prior G2 CAR T cell trial. So this is like a normal CAR T cell. So that has signal 1 and signal 2. So that's kind of what people use for like all the FDA approved trials. So you can see like some a line of purple. So this is like the CAR T cell numbers with chemotherapy. So you can see with chemotherapy, the CAR T cell numbers kind of go up here and then kind of start going down. So clearly like the chemotherapy is needed. And so if you don't have the chemotherapy, so like this is like the black line here. So this is like the 4-hour level. This is essentially like the initial CAR T cell level right after infusion. So you can see here, this is 4 hours, and like compared to at week 1, you actually see like the line is actually going down. So it means like between like 4 hours and week 1, these cells are actually not expanding at all, and some of them actually dying off. So that means like without the chemotherapy, with like a normal CAR T cell, these cells just don't really grow and actually like die off over time. So again, I think the most important difference is like with this C7R, you can see that we treated 3 patients without the chemotherapy, and you can see actually like the numbers actually did go up even without the chemotherapy here in black. So that indicating that you don't need the cytokine that is made by the chemotherapy for these T cells to grow. So what does that mean in terms of like the clinical kind of picture? So first, like what do we see in terms of side effects? So yeah, we did see cytokine release syndrome in 8 of 13 patients. We didn't see any cytokine release patients in the 3 patients with lymphodepletion. So we again, something potentially interesting. So even though they had very similar expansion, we actually didn't see cytokine release in the patients who didn't get chemotherapy. But most of the patients who got chemotherapy got cytokine release syndrome. Early on, so the standard of care is to use tocilizumab to treat cytokine release syndrome. So that's what we usually use for all of the FDA-approved CAR T cell treatments. But we actually found that actually wasn't really that effective to use tocilizumab. So after we kind of looked a little bit closer to like and what's happening in these patients, we saw that a lot of these this inflammation was probably a myeloid cell. So it's like a different types of immune cell that was causing the inflammation, not the T cells themselves. So we target these myeloid cells with a different drug called anakinra. And once we started using this anakinra earlier, we actually saw that we were able to control the CRS much better. So we've been much more aggressive with using anakinra, and that actually has resulted in like not getting the really kind of severe cytokine release syndrome that we saw in some of the patients. We didn't see any, so ICANS is a type of neurotoxicity that's commonly seen with CAR T cells. So we didn't see any neurotoxicity, and we didn't see any of the prolonged cytopenias that we see with other types of CAR T cells. A lot of leukemia patients get, very severe neutropenia for a long time, which can cause infections and other problems. So it can take a couple of weeks for the counts to recover in some of the patients, but we haven't seen like the kind of month-long clinically relevant cytopenias that cause infections in our patients. So, like I mentioned before, we do think that using the anakinra earlier results in less toxicity, and has been helpful.
Dr. Omer, so I know you don't really want CRS, but have you seen any correlation between the degree of CRS with response? Just because it's one of those things where you're like, is anything happening in there? So any correlation between that?
Yeah, I think it's a good question. I think, early on, thinking with the CAR T cells, people say, well, if you don't get a fever, it's not going to work. I think it's not really quite like that anymore. I think one factor is like how much disease there is. So there's like a lot of very high tumor burden, very big tumors or a lot of tumors. That means there's more antigen. So more antigen leads to more inflammation. More inflammation means more CRS. So patients who have a lot of disease do tend to have more cytokine release syndrome. If you have like very little disease, there's like less inflammation, so there's like less cytokine release syndrome. And the second factor is like the dose. The higher doses, we started seeing like a lot of inflammation. And I think one problem is like that if you get too much inflammation, I think that these cells just kind of start flaming out to some degree without really kind of doing what they're supposed to do, which is kind of going to the tumor. So I think some CRS is good, but I think if you have like too much CRS, it's probably not say a good thing, because like the T cells just you kind of get the cycle of inflammation, and it's not necessarily kind of like like the good kind of inflammation. Yeah, I don't think that's like a straightforward answer, but I think they're cytokine release syndrome to some degree, I think it indicates that the T cells actually like going to the tumor, recognizing tumor, secreting substances that cause inflammation. So on one side is like a normal response. On the other side, maybe too much of that is probably not necessarily a good thing. And I think the reason like the inflammation is high is probably the combination of the chemotherapy. So we know, so if you give the chemotherapy, there's kind of like a rebound. So the bone marrow is empty, so the bone marrow kind of starts making more myeloid cells, and it's like a known phenomenon, and people call it reactive myelopoiesis. So like it's like a reaction essentially to the chemotherapy. So if you give the chemotherapy, you have this kind of myeloid inflammation just from the chemotherapy itself. So then if you add the CAR T cell to that, you kind of supercharge that inflammation from like the chemotherapy. And while the T cell inflammation is like a good kind of inflammation, like the myeloid cell inflammation is not it's not a good kind of inflammation. So one way of targeting is anakinra. But a second way, and I'll kind of come back to that a few slides from now, is to maybe actually take out the chemotherapy again and say, well, it doesn't seem like you really need the chemotherapy for like the T cells, because we already have the cytokine supply kind of built into the T cells. So if you take out the chemotherapy, you don't really get this myeloid inflammation from the chemotherapy. So you don't kind of have this like kind of double whammy of like inflammation from the chemotherapy and inflammation from the T cells. So I'll talk about that a little bit more in a few slides. So these are the responses here. You can see the partial responses are all in neuroblastoma patients. The patient had progressive disease. So neuroblastoma had a patient had like very, very, advanced disease with like tumors essentially like all of their body. Again, kind of indicating what I mentioned before. So if you have less tumor, you tend to have better responses. And like some of these partial responses actually lasted pretty long. So this is a 7-year-old patient with neuroblastoma. She actually had a partial response to her initial infusion with lymphodepletion. So she had a rib tumor. I have the picture here. This is like the picture before, as a picture after. You can see like the rib tumor resolved. She also had an abdominal tumor that also resolved. And then there was a little area left in the leg. So we actually gave her another infusion 3 months later, and that time without lymphodepletion, based on a couple other factors. And then the patient had just this very tiny area of tumor left for about 8 months, which stayed stable. And then unfortunately, after 8 months, this tumor started coming back again. And we have one advantage with neuroblastoma which we don't have with osteosarcoma, which is we have like a nuclear imaging test. So that kind of helps us to actually look at like the tumor that's actually viable or kind of active. So if you look here, so this is like an MIBG image. So this means like the kind of yellow signal here is kind of like active tumor. So the active tumor, you can kind of see it disappears here. But if you actually look at the CT scan, you actually wouldn't have seen a difference. So I think neuroblastoma is a little bit easier in that we can actually see, okay, the active tumor kind of disappears, so it gets less. But with osteosarcoma, we don't really have that ability.
That's amazing, though, that there was that, response for 8 months.
Yeah.
But I'm curious because you gave a second infusion after 3 months. So what are the thoughts about giving, additional infusions every 3 to 4 months? Because that would have, would that have prevented the recurrence at 8 months?
Yeah, something that's a good question. So we have a separate brain tumor trial, and we've seen that patients start. Patient have a response for about 6 to 8 weeks, and then the tumor starts coming back. So what we've started doing is we kind of give infusions every 6 weeks. So we, give infusion. After 6 weeks, give the next infusion. And this is still pretty early on, but like it seems like we can extend these responses by kind of giving like repeated infusions. So we just started doing this in a brain tumor setting, but yeah, I think that's something that's potentially a consideration. And it's a little bit hard, because a lot of the patients are from out of town, and like the way we've written the protocol, you have to be in Houston after infusion for at least 3 weeks. So it's a pretty big commitment, right? So you have to kind of come here, stay here for 3 weeks, and then if you do another infusion, you have to kind of come back here again. So we started doing that for the brain tumor patients, mostly for the patients who are a little bit kind of in Texas, so it's a little bit easier for them to kind of get here. But yes, it does seem like we can kind of extend like the responses if you kind of give these infusions every 6 weeks. So this patient here is a patient with metastatic osteosarcoma. This patient has very rapidly progressive disease. It was chemo refractory. So this patient actually did get lymphodepletion. You can kind of see the tumor here in the mediastinum, pretty big. It was actually a bit higher than like the threshold for like what we're actually allowed on study, which is like 5 centimeters. But you can see that post-infusion, you can see like these kind of black dots. There's some necrosis, probably likely dead tumor tissue, and then like the overall size of this actually has decreased. And again, it seemed like this patient responded for about 2 or 3 months, and then the tumor started progressing again. And the circle just kind of shows like the area where I think there's kind of necrosis happening. And then this patient was interesting. Was like this patient had a sodium fluoride scan. So usually we don't get sodium fluoride scans, but like sodium fluoride scan is essentially like a way to kind of look at osteosarcoma that's kind of active. So on the CT scan, we actually didn't see any response. But then if you look at the sodium fluoride scan, you can see here like these black dots, kind of like active tumor here. So I just kind of highlight like an area here, so you can see like, you know, there's like a pretty large area of tumor here, and there's kind of like, you know, quite a bit of tumor here, kind of in the lungs and the ribs. You can see like post-infusion. So this is week 5. You can see, you know, this big area is a lot smaller. And you can see like, you know, some of these areas actually have resolved. So this is like 5 weeks after treatment with no other treatment. And then, you know, similar, you know, this patient was pretty similar. So like this lasted for, you know, about 2 months, and the patient progressed again. So I think in summary, so we do see CAR T-cell expansion and persistence even without chemotherapy. We haven't seen any cytokine release syndrome or any really toxicity without chemotherapy. But we have seen a lot of kind of grade 2, 3 cytokine release syndrome with chemotherapy. We did find that there was better control of the cytokine release syndrome when we use anakinra first line for cytokine release syndrome. And we had partial response or stable disease in 8 of 11 patients treated post-infiltration. And actually, a lot of these stable disease patients, not all of them, but like some of them actually did have improvement in some areas of their tumor. But again, to really qualify for partial response, the criteria are pretty strict. I mean, you know, for example, if a lot of your tumor improves, but there's like 1 new area, then, you know, it's not a partial response, progressive disease. So there's quite a few patients where, you know, things actually did look better for some time, but they didn't. They still didn't qualify for anything better than stable disease. And I think one thing I've noticed, you know, for example, patients who have pain from their tumors, you know, we've got a couple of patients who had significant pain. So patients who have pain, they do have improvement in their pain, even with stable disease. And I think to me, again, this is like an indication that, you know, we're seeing some tumor response, and that, you know, you have decrease in pain, probably decrease in some active tumor. But again, with a single infusion, you know, these, you know, we're talking about like, you know, response lasting like 2 or 3 months. And I do think that the CT scans may underestimate the response. You know, like I said, like in neuroblastoma patients where we do both CT scans and DIPG, we often don't really see much of a response on CT scans, but like we see an improvement on the DIPG. So what we're going to do, you know, in the future. So initially we thought, okay, well, we probably need the lymphodepletion, even though these CAR T cells have the cytokine supply. But as I just showed, we actually get pretty good expansion without lymphodepletion. So I'm actually just working right now on modifying the protocol to maybe just try a higher dose again one more time without lymphodepletion. So the thought being that we can maybe push the dose a little more if you don't get the cytokine release syndrome, and maybe get a little bit less of like this myeloid inflammation. Again, we don't know if that's going to work, but I think we have, you know, I think the data kind of indicates that, you know, it's at least it's worth a try. And with the advantage of that being is like you don't need any additional chemotherapy. You know, there's no toxicity from the chemotherapy itself. So potentially, you know, in the future, this might be, you know, if you can get the same results with just the T-cell infusion without any kind of toxicity from the chemotherapy, this might potentially be like something you can kind of use as a bridge, kind of like as a break from chemotherapy, where you can maybe just kind of control the tumor for a couple of months at least, and without any kind of you know, bone marrow toxicity or things like that. The initial patients we treated without lymphodepletion, except for the one patient, didn't respond, but these patients also had very, very big tumors. So I'm talking about, you know, 10 centimeter, 10, 12 centimeter tumors. So it's very hard to kind of really kind of get a response if your tumor is, you know, is this big. And like I mentioned, we didn't see any cytokine release in none of these patients. So we have an ongoing brain tumor trial with the C7-G2 CAR T cells with promising results. And I'm mentioning it because like this trial is interesting because we're actually directly comparing the first 3 patients only got G2 CAR T cells, and the patients after that have been getting the G2 CAR T cells with the C7R. And we saw longer lasting responses with the C7R. So like without the C7R, the responses only lasted about 3 weeks. And with the CCNR, we've been seeing responses that last, that have been lasting 2 months, 3 months, 4 months, and are still ongoing in some of these patients. So indicating that the CCNR really indeed does kind of improve the G2 CAR T cells as we think. And on that trial, so 3 of the 3 patients we treated with the CCNR actually had clinical improvement. All of these patients actually had symptoms from their brain tumors, and we've actually seen pretty minimal toxicity with these tumors. So again, I think indicating that the CCNR does actually do what we're kind of hoping to do, which is kind of extending the duration of the G2 CAR T cells. And that's it. So I just want to thank, Dr. Rooney. She's my mentor. She's been, working on CAR T cells for a long time and T cell therapy. She was one of the first people to actually use T cells to treat cancer. And so she's one of the pioneers of the field of T cell therapy, and actually, pretty famous. And then Thomas Shum, so he was a grad student who worked with me, and Dr. Rooney. So he actually had the idea for the CCNR, after a lot of kind of brainstorming sessions, kind of late at night, and so he actually kind of came up with this mutation. So he kind of moved on. He's now at Harvard. So I want to thank him for his contributions, and of course thanking all the patients that have enrolled on this trial, and especially early on, it's scary because you don't know what to expect and kind of what's going to happen. And then the funding for this trial was from the Cancer Prevention Research Institute of Texas. So of course, if you don't have money, then we can't do the trial. And then all the co-investigators, statisticians, study coordinator David Allen, him, and then the people in the lab kind of developed this, and our regulatory team. So and then again, thank you one more time to allow me to kind of share our experience. And I'm happy to take any additional questions.
Thanks so much, Dr. Omer. Please feel free to put questions in the Q&A or even chat, and we'll look out for those. And then I think, Matt, you had a question.
Yeah, I had a question about, hold on one second. I guess you were kind of going into this a little bit at the end there, but how long exactly do the T cells need to endure in the body for there to be an optimal response?
Yeah, I think it's a good question. I think we, for example, in leukemia setting, we know that, if they can stick around for 6 months, then, 6 to 12 months, then your, the chance for a cure is pretty good. So the longer the better. We don't know kind of what the duration is really kind of for osteosarcoma. I think in general, I think the problem is if you don't really mop up every last single tumor cell, there's a chance that the tumor can come back, right? So that's kind of the tricky part. So if you're not able to kind of get rid of all of the tumor cells, or main tumor cells, then it's easy for the tumor to come back. So there's some new approaches, where people kind of using essentially mRNA vaccines to boost CAR T cells. So I just saw some data from BioNTech, which developed the Pfizer COVID vaccine. So they're using one of their mRNA vaccines to boost CAR T cells, and this is a solid tumor trial for actually ovarian cancer. So they're actually able to kind of boost the CAR T cells. So instead of giving repeated doses of the CAR T cells, they actually give they give this vaccine, and the vaccine kind of boosts the CAR T cells. So they kind of keep giving these vaccines to kind of extend the duration of how long the CAR T cells work. So I think that's something kind of for the future. I think for now, I think kind of like Christina suggested, I think the best approach might be to just kind of repeat infusions. And like I said, in the brain tumor setting, we have some data that indicates that it actually does seem to work, where it can, kind of keep chipping away at the tumor, so to speak, where, with each infusion, you kind of get, some of the tumor kind of at least gets controlled, and maybe some of the tumor can kind of shrink. So the thought is maybe kind of keep chipping away at the tumor with kind of multiple infusions.
Okay, interesting. Thank you. And we just got another question from someone in the audience. They said, could or would this be a treatment used for currently non-metastatic or patients currently NED as a treatment to proactively kill any undetected tumor cells?
Yeah, I think that's very intriguing because I think I personally think it's much easier for T cells to kind of mop up a couple kind of stray tumor cells and kind of like tackle like a big kind of bulky tumor. So I think it would be very interesting kind of use CAR T cells maybe like similar to kind of what people use antibody therapies for. For neuroblastoma, people use G2 antibodies to kind of as like an adjunctive treatment. Or if you just use antibodies, they don't really do anything or don't do much. But if you kind of use the antibodies after like all your other kind of chemotherapy and treatments, you can actually get like a higher cure rate than if you don't give the antibody. So I think CAR T cells might be able to do something similar. I think for now, the FDA is not going to allow you to use it in patients without any active disease. So we are allowed to use it for patients who don't have metastatic disease. So if a patient has a recurrent tumor and like, the primary area of disease, so that would be acceptable. And then if patients have no disease, what we have done before is again something we kind of mentioned earlier. So what we've done before is we've made the CAR T cells, or at least kind of collect the blood, and then made the CAR T cells. And then have the CAR T cells, and then there's been like at least one case of a patient where they then emailed me like a year later, like, oh yeah, we just unfortunately like we relapsed, and so yes, we can potentially make it the CAR T cells before relapse, so like when the patient's like NED. But we can't infuse it unless there's like at least some measurable tumor. And again, this is kind of based on the FDA. There's a pretty high threshold to kind of use like these types of therapies. And I think that might change in the future. And we now have like a lot of evidence that CAR T cells are relatively safe. Yes, and we kind of get the cytokine release syndrome, but we don't have any all these patients have completely recovered, and there's been like no kind of long-term kind of effects from these CAR T cells to my knowledge. And we've treated now across like different institutions, we've treated probably a couple hundred patients. So I think there's enough of a track record that you can make an argument that maybe this can be kind of used like in the adjunctive setting where you just kind of do some other treatment or kind of do surgery and then kind of get the CAR T cells to kind of maybe increase your or decrease your risk of recurrence. So yeah, I think it's a really good, really good question.
Yeah, thank you for whoever asked that question.
That was a really good question, because I was just thinking, we've had previous guests talk about different biomarkers for risk stratification. For example, I think at this point we know MYC is a pretty good biomarker for high risk. And so imagine even if you were NED, I know this is a trial, so you need to have some visible disease so that you can measure, but this would be great to do after MAP, for example, as kind of a preventative for high risk. And Dr. Omer, you're currently recruiting still, right, for this trial?
Yeah, so we're still recruiting. I'm actually just working on this amendment to kind of take out the chemotherapy, because I'm really kind of intrigued at I kind of want to see if you can, if you just use the T cells, could it kind of boost the doses without really getting a lot of toxicity. I'm working on that now. So it's probably going to take, usually takes about like 2 months or so kind of for a protocol to be approved. So we can still, collect, we can still kind of, do the testing and kind of make CAR T cells. It's probably going to take at least, about 2 months where we can kind of treat like the patient without kind of the chemotherapy. But yeah, I think that's kind of the plan right now. And I'm just drafting the letter to the FDA. So hopefully I can get this in either this week or next week.
Okay, great. And then what we'll do maybe is we'll share your contact information for those folks registered for this OsteoBite, so that if they are interested in getting more information, they can get in touch with you. And thank you so much. So interesting. And we've come to the end of our hour. So I just want to thank you so much for taking the time to join us today, Dr. Omer on Osteobites, and for making it better for pediatric oncology patients everywhere. More information on this and all osteobites can be found on YouTube, on our channel, at our website, mibagents.org, and your favorite podcast place. And next week, we are talking with Dr. John Healy, the Chief of Orthopedic Surgery at Memorial Sloan Kettering Cancer Center. And Dr. Healy will be joined by osteo warrior Brandon Friedman, and together they will offer perspectives on limb salvage surgery for osteosarcoma. So thanks again, Dr. Omer and Matt also for spending an hour with us today. And of course, to our sponsor, BTG Specialty Pharmaceuticals. And thank you all for joining us today on Osteobites. We hope to see you next week when we chat with Dr. John Healy and Brandon. Thanks so much. Have a good rest of the week.